Imagine you're reading the instruction manual for building a human, and 98% of the pages are blank. This is what scientists found in 2003 after sequencing the human genome, expecting over 100,000 genes, but finding only about 20,000. This left a huge question. Where is all the extra complexity hiding if not in the genes? It's hidden in what was once dismissed as junk DNA, a label we're now removing forever. For decades, scientists like Susumu Ono believed this extra DNA was useless, a graveyard of broken genes. But then, two massive projects changed everything: ENCODE and Genome-Wide Association Studies, or GWAS. ENCODE revealed that at least 80% of the human genome is biochemically active, not silent. And GWAS studies found that over 90% of disease-linked genetic variations live in these so-called junk regions, not in protein-coding genes. This was the smoking gun. The silent stuff isn't silent at all. Your genome is not a straight line. It's a 3D masterpiece, folding like intricate origami to fit inside a tiny cell nucleus. These precise folds bring distant silent regions, called enhancers, right next to the genes they control, acting like remote-control dimmer switches. This is why you can speak, but a chimp cannot. Despite sharing the same FOXP2 gene, your enhancer network is far more elaborate. Beyond DNA, non-coding RNAs are the real bosses, like microRNAs, tiny assassins that shut down hundreds of messenger RNAs. Then there are long non-coding RNAs, or lncRNAs, the architects. Xist, for example, coats an entire X chromosome, zipping it shut into a silent bar body. Even ancient retrovirus fossils, making up 45% of your genome, have been domesticated by evolution, now controlling vital functions like placental development. On top of your DNA sequence sits epigenetics, chemical tags that act like highlighter marks or sticky notes, telling your cells what to read and what to skip. These tags, like DNA methylation and histone modifications, land mostly in the silent regions, differentiating a liver cell from a neuron despite identical DNA. Remarkably, some of these epigenetic marks can be influenced by your environment and even passed down through generations. New tools like next-gen sequencing, Hi-C, and CRISPR screens are finally allowing us to see and understand these previously hidden genomic layers. This understanding is revolutionizing medicine, as most genetic risks for diseases like cancer and Alzheimer's come from broken volume knobs in the silent 98%, not just broken proteins. Future drugs will target these broken switches, using technologies like antisense oligos or epigenetic CRISPR to rewrite the actual operating system of our bodies. The genes we obsessed over are just the parts list. The real magic, the reason you are uniquely you, lives in the silent 98%. It's the enhancers, the lncRNAs, the 3D loops, and the epigenetic marks. The operating system evolution spent 500 million years debugging. We used to think the genome was a book. It's actually a symphony, and we're finally learning to hear the quiet instruments that carry the whole melody. If your mind is blown, hit that like button, subscribe, and ring the bell for more insights into the hidden wonders of our biology. Thanks for watching. Keep looking up and inward. See you in the next one.